Good afternoon and welcome. My name is Christine Russell, and I'm the Executive Editor for Medical Laboratory Observer. Our speaker today is Rob Andrews, Flow Cytometry Director for InVivoScribe. Robert has worked in the clinical flow cytometry field for the last 12 years in varying institutions such as Genoptics Medical Laboratories, BD Biosciences, and LabPMM. Robert has a passion for helping others in the flow cytometry community, and over his career has aided in the setup of multiple flow cytometry labs across the U.S. for leukemia and lymphoma immunophenotyping. Currently, Robert is directing the flow cytometry lab at LabPMM in San Diego, California, specializing in MRD testing for AML. Our webinar today is titled A Streamlined Clinical Lab Workflow for Controlling Antibody Reagent Lots on the 12C BD Fax Lyric Flow Cytometer. We have a few housekeeping items. In order to submit a question for our presenter, please use the Q&A box at the bottom left of your webinar window. For tech issues, please click F5 on your keyboard to refresh. If that doesn't work, submit a question through the Q&A panel. Robert, we look forward to your presentation.
Hello, everyone. My name is Robert, and I would like to thank you for joining today. I hope you find some of today's information useful and can apply some of these ideas or tools to your laboratory workflows. Currently, I'm the director at LabPMM, a fully owned lab of InVivoScribe, Inc. I have no disclosures to announce for this presentation. A little bit about LabPMM. We have implemented all that we'll be discussing today in our flow cytometry lab. LabPMM is a globally standardized network of specialty reference labs that provides best-in-class NGS assays, gene panels, and flow-based assays, including rare event analysis. We provide our partners with the unique solution of having multiple complementary technologies on the same pool of bone marrow so that all testing can be performed within LabPMM. By offering both flow and NGS-based testing, we are able to accelerate clinical trials and streamline drug approvals. Recently, we just launched a standardized 12-color AML MRD assay by flow cytometry and also help our partners develop CDX assays across our multiple testing sites. I work in the San Diego lab, but we also have standardized labs in China, Japan, and Germany as well. So today, we'll be going over learning quality control procedures to meet regulatory compliance in a laboratory setting, such as CAP checklist items. Streamlining laboratory workflows to meet these amongst busy specimen processing labs using instruments and analysis software, and ensuring correct implementation of lot-specific spillover values in your multicolor antibody cocktails using the Fax Lyrics and Fax Suite software. CAP checklist items are a great laboratory practice even for labs not accredited by CAP, helping standardize processes and assure continuous quality. Summed up, we will cover processes to assure received antibodies and buffers are qualified before use, and that concentration is matched between your antibody cocktails and what is applied to your multicolor assays. I have worked in a high-volume CAP CLIA flow cytometry lab over the last 12 years and know the time constraints on expected turnaround times, often with clients wanting results on the same day. Flow labs, including global clinical research organizations and reference laboratories, are faced with complying with regulatory standards, including CAP checklists. During this presentation, we will not only address the above checklist items, but solutions will be provided to meet these requirements in a streamlined fashion. Using automated and semi-automated software techniques will help reduce time and error associated with antibody QC and reagents. The three main requirements that we'll focus on today will be checklist items flow 23275, antibody validation, that the laboratory has recorded the initial validation of new reagents before use. Flow 23325, new reagent lot shipment confirmation of acceptability, essentially that new lots and shipments of antibodies and licensing reagents are compared with old lots before or concurrently placed into use. And flow 30260, that color compensation settings are established for determining appropriate spillover, frequency, and assessment of those settings. The workflows covered will just be some ways to do this in labs. There's also other ways that can be done as well. We use a combination of internal forms FCS Express analysis templates, and fact suite to accomplish this in our lab. So starting first, single antibodies. Some will say that these are the most important reagents in your panels. In our lab, before antibodies can be used, they must be qualified. We use antibody titration experiments to assess antibodies that come into our lab for specificity and reactivity before use in multicolor assays. Antibodies are assessed by the use of known positive and negative control donor cells stained at varying concentrations and then analyzed to determine expected reactivity and optimal titer. A titration assay is used to verify antibody reactivity and specificity and also to determine the antibody concentration resulting in the lowest level of nonspecific binding. Not using enough antibody can usually result in lower MFIs of positive populations and decreased resolution from dim positives and negatives. Too much antibody can cause binding of antibodies to low affinity targets, which can increase background fluorescence. All titration assays are performed under the same experimental conditions as the assays the reagents are intended to use for. Examples, if you're running, an assay that has 1 million cells stained versus 5 million cells stained, you'll want to titrate your antibodies in the appropriate conditions, including number of cells and total reaction of the experiment. We use this example here, an automated FCS Express template that calculates the ultimate titer based off of stain index. Essentially, we run serial dilutions, 1 to 2 serial dilutions, usually starting at the manufacturer recommended concentration, and then export the files from fact suite into this template, where the gates are adjusted first around the singlet population, then around the cells of interest, then the specific populations are gated based off of the marker that you're looking at in expression, and then the stain index is calculated based off of the tokens in the work list. This template will provide us with the calculated data, the plot, and also the optimized titration on the top, all automatically. All of our antibodies are qualified in this manner before they are used in assays and validated within the assay before used on any clinical samples. Usually, starting volumes will look to manufacturer for guidance on the TDS and take into account the cell type, quantity, and reaction volume. This is a great way to optimize not using multiple softwares to calculate the titration for your titration experiments. Once we've qualified our antibodies that are used in our lab, we lot-to-lot parallel test each new lot or shipment of antibody. We use this form to document the information we need to place the reagent into production. This helps all of our technicians, once we've qualified labs that we use on antibodies that we use on a regular basis, to keep track with what antibodies have been lot tested and what information that they need to perform it. As new lots and shipments of single antibodies are received into the lab, we first check the TDS and check for any updates, and the antibody information is then added to the log, including specificity, dye, manufacturer, catalog number, lot, and expiration date. The current lot information is also obtained from the current antibodies in the lab and entered into the parallel testing document here. This log will allow the technologist to track which antibodies need to parallel QC, and once it has passed, they can move these lots from quarantine to production. This form is used in combination with our single antibody parallel QC template for each individual antibody and are filed in our antibody QC binder. This log allows all the technologists to know what reagents need to be QC'd and saved on the net, and then we save these on the networks. We also add what settings need to be updated for spillover values to this form based off the conjugate, and we can parallel test and set up lot-specific spillover for each new reagent as we parallel QC them. This form saves time in keeping everyone on the same page. Now that we have covered antibody qualification and documentation of receiving shipments of new lots and shipments, we will discuss how we assess the performance of each new lot. This covers the flow checklist item, flow 23325, performance of new lots and shipments of antibodies. This also includes cocktails, which should be compared to previous lots before going into use. The performance of new lots and shipments of antibodies and reagents is compared with old lots before concurrently being placed into service and the records are retained. Flow cytometry assays are developed and optimized with specific clones and conjugates of antibodies directed against specific biomarkers. These antibodies used in flow assays need to produce a specific and consistent single on negative and positive cells of interest. Since expression levels of antigens on cells help identify aberrancies, it is important that intensities of antibody conjugates are consistent over time and from lot to lot. When developing assays, antibody titration experiments are run to determine the optimal concentration of an antibody for the particular assay. Then with new shipments and lots of antibodies, verification of consistent intensity and optimal concentration must be documented so that the assay results and interpretations can be standardized. By parallel staining new lots and shipments of antibodies with defined acceptance criteria, comparable results can be standardized over time and across reagent lots. In this example, normal peripheral blood is stained with 2 different lots of BB605CD5 and run on a 12-color fax lyric. The data is exported into FCS Express and gated. Tokens in the layout calculate the MFI difference between lots and flag pass-fail. Our acceptance criteria is less than or equal to 20% MFI between lots. Also, the staining patterns must be comparable. When lot-to-lot QC fails, usually the setup is reset up or is retitered. In this template here, we enter all of the lot information for the reagents being tested, stain appropriate samples, and then acquire the data. First, we gate on the singlet cell population, then the population of interest for the biomarker being assessed, and then the gates for on the histogram. The gating is linked between the current and new lots, and then the template updates the median MFI of the current lot and the new lot, and then calculates whether it's within our 20% MFI cutoff acceptance criteria. Expected staining pattern of the new antibody is assessed as well to make sure it's working appropriately. If the antibody fails, it is usually reset or retitered. 9 times out of 10, if it fails, the new lot is too bright, and the retitration allows us to use a different volume possibly. A similar approach is used for reagents such as buffers as well. Confirmation of new lot acceptability not only applies to antibodies, but lysing reagents as well. Verification of performance and qualifying of critical reagents prior to testing is a fundamental requirement to achieve continuous quality throughout the flow cytometry testing process. QC testing of lyse reagents should be conducted and verified prior to reagents being placed into use. Last year, a new checklist item was added, lot-to-lot parallel QC for stock lysing solutions. Each new lot of 10x lysing is parallel tested in our lab on the same sample as the current lot. Acceptance criteria includes comparable light scatter characteristics, staining patterns, viability, and percent debris between lots. In this example, a single multicolor tube is acquired and processed on a 12-color fax lyric. Data is dropped into an analysis template and gated. Tokens automatically populate comparison tables to determine if the new lot is acceptable. The criteria assessed was chosen by our lab to confirm that the lysing reagent is performing, but also not producing any interference with reagents or harming cells. The buffer is tested at the 1x concentration, and we compare the lot, debris, viable cells, and light scatter. So here you can see a multicolor assay of normal blood setup where we gate singlet cells, gate leukocyte populations, and then look at the expression of normal markers on the cells. We compare the intensity of the MFI between the different populations, the percentage of cells, the percent debris, and the viability of the sample. The tokens automatically update based off of the gating. Weekly, we perform a 1 to 10 dilution of that 10x stock lysing buffer to a 1x working solution with reagent grade water. The buffer is qualified before use as well. This qualification ensures all critical components of the testing process are qualified before used on samples. The same process is performed for wash buffer used in our lab so that all buffers are qualified before use. A form is used to document lot-specific and preparation-specific information, as well as documenting of passing of QC or not. On that form, we have all of the ingredients that go into the lot including raw materials and a basic description of the preparation process. A buffer qualification assay is used in the work list of the fact suite, and expressions calculate the percent of viable cells and debris from the gates in the report. The assay is saved in the library for ease of use and is updated daily with the running of assay tube setting setups. This allows for ease of use and reproducibility in the buffer qualification workflow. The same assay is used also for our wash buffer. Something to note is that the pH is adjusted for both buffers between 7.20 and 7.40 before testing. We also filter our wash buffer with 0.2 micron filters if needed. Performing this on our wash buffer and our lice buffer helps us control our testing environment and make sure that all of our reagents in the testing process have been qualified before use. We make our own buffers in-house, but the same testing mechanism and workflow can be used for commercially bought buffers such as BD stain buffer or BD farm lice. Now that we have covered single antibody QC and buffer QC, we will move on to antibody cocktails and compensation. This will cover the flow checklist item 30260. Procedure should be established for determining appropriate color compensation settings. Compensation needs to be addressed when one or more fluorochromes are used in an assay, in which the overlapping spectral emission with another fluorochrome cannot be addressed with the filters of the instrument alone. This spectral overlap causes a primary dye to leak into a secondary dyes detector and is called spillover. The mathematical subtraction of the primary dye leaking into the secondary dye is called compensation. Spillover values are calculated by the running of single stain controls and are computed by the instrument software. Spillover values are established either by the running of BDFC beads, single stain capture beads such as BD comp beads, or single stain cells stained with one dye. Once calculated, spillover values are automatically updated during daily instrument QC. Spillover values need to be updated based off the laboratory's validated protocols. In this example, we can see spillover of PITC into the PE filter and a little bit of the PE filter spilling into the PITC filter. That spillover is what is being compensated or subtracted out so that the signals inside are only coming from the primary dye. In this slide, we'll address the workflow that we use for addressing lot-specific spillover using the FACS Lyric flow cytometer. Essentially, first we update our library with new labels and reagent lot information as needed when antibodies are received. We've covered this, how each new lot of reagent is parallel tested. At that time, if the parallel tests pass, we also update the lot-specific spillover values and add them to the particular reference setting associated with them, such as lice wash. We run lot-specific single stain controls during this time for mostly tandem dyes, new dyes, or dyes that need to be stained with cells. Next we create multicolor cocktails and document the antibodies used in batch records. Then we run fully stained samples with the current cocktail lot and new cocktail lot and compare the MFI of lots and compensation of the new cocktails. If this passes, we evaluate compensation of each run sample and document for adequacy on each sample analysis report. That's done on a daily basis for each sample run. Every 60 days, we run FC beads to update the lot-specific spillover, or update the FC bead spillover values in lice wash, and run lot-specific controls that we've done with the add fluorochrome function every 60 days in the library, which we have validated in our own laboratory setting. Now we'll go into the more nuts and bolts of how we do this and a workflow that we have found works well for us in our laboratory setting. Now we'll start with the steps we perform to add lot-specific spillover values for conjugates that we need. This takes place during the single antibody parallel testing run, which also is when we add lot-specific spillover values for reagents such as tandem dyes and other reagents that we have found that need it, such as BV510 in our experience. In our library, we add the antibody conjugate lot and expiration information, and then later match the measured spillover values in the library assay settings work location. Ultimately, the correct lot-specific spillover values can be applied to the correct multicolor experiments. Once the information is added, the spillover values can be calculated after any time. Here in the library section of FactSuite software, we add the new reagent lot and expiration dates. For rare antigens, you might first need to add the label in the label section of the library. Here's a snip of some of the reagents that we've added for various panels, showing the lot information and expiration date documented in the software. This information can be stored and then later removed once the antibody is no longer in use. The information in here can be paired to any assay created and the properties of the assay so that lot-specific compensation can be performed. Also, you must add the label and then the reagent before you can add any of the spillover values for a particular reagent for lot-specific compensation. This is all done in the library function or the QC function of the FactSuite software. Once the antibody label and reagent information is entered into the software library, single stain controls can be set up and run for lot-specific reagents. For lot-specific comp, either cells or beads can be used. We use both. In the setup and QC module, we choose the add fluorochrome option, choose the correct reference setting to add the lot-specific spillover value to, choose the control type, label, and lot, and then run the control to measure spillover value. In this example, CD19 PE-Cy7 is added to light wash reference settings using BD comp feeds. Once the lot-specific spillover values are calculated, they are saved and can be applied to any experiment using the reagent conjugate lot. These saved spillover values will adjust automatically based off the tube settings they are applied to and saved in the light wash or light snow wash settings. If they're applied to a particular assay during the assay tube setting setup run in the morning, a report will be executed and you'll see the updated spillover values based off of the MFI targets. A little bit about antibody cocktails. Antibody cocktails are solutions made of individual antibodies mixed together, usually with cell wash buffer containing PBS, BSA, and sodium azide. Brilliant Stain Buffer Plus is added to cocktails when more than 1 billion violet dye is used. We cocktail in our lab up to 12 colors using 4 to 5 billion violet dyes. We usually have a stability of 3 weeks to a month with these. Cocktails combine antibodies that are used together in the same tube and provide several advantages, including increased efficiency by reducing the number of pipetting steps, decreased errors and variability by eliminating the need for pipetting small volumes, and increased quality assurance by verifying the presence of antibodies even when the antigen of interest is not present in samples stained with the cocktail. Antibody cocktails also provide an ergonomic solution to the high volume of pipetting steps within the flow cytometry lab process. In combination with electric pipettes and multi-dispense features, the volume of pipetting steps can be reduced more than tenfold. We currently not only work with liquid reagents in our lab, but are also optimizing dried down antibody technology with the BD custom reagents team. Cocktails must be parallel tested lot for lot as well, and also have stability tested when validating in new panels. When first creating cocktails, they should also be compared against single stains to make sure that the MFI the antibody combinations aren't resulting in significantly decreased MFI for any of the reagents. So for each new cocktail, you should have a batch record. The batch record documents the antibody conjugates and lots used in the cocktails that will later be applied to the cocktail-specific assay in FactSuite. Having this information will help you prepare your new assay in FactSuite so that lot-specific spillover value can be assessed. The technologist who prepares the cocktail in our lab verifies all reagent lot information and lot-specific spillover values are added to the library and then creates a new copy of the current assay with the new lot information and spillover values assigned. The assay will be specifically named with the cocktail prep date in addition to the assay name. Once cocktail parallel QC is run, the approval of meeting the acceptance criteria for the cocktail is documented on the form, and as well as the date and time of going into use. In our batch record, we list the date prepared, expiration based off of validated sample stability, initials of who prepared the cocktail, the defined lot number of the cocktail, which we go with the date the cocktail was made, list of all antibody conjugates within the cocktail and their catalog number, and the lot and expiration of each reagent. Also incorporated is a place for sign-off of cocktail QC passing when the cocktail has been put into use, and the sign-off of checking that the new cocktail assay has all the correct lot-specific spillover values assigned to it. So once the cocktail is prepared, we must create the assays and fact suite to parallel test against the current lot. We create a new assay off the current assay and name it with the new cocktail date. In the library of fact suite, you can see here in the slide that you can assign either generic or lot-specific spillover values to each conjugates in a panel. In this panel, most reagents are run with lot-specific compensation to provide the highest quality of data possible. The lot numbers assigned to the assay are based off the cocktail batch record. When preparing a new cocktail, we run the current cocktail with the correct spillover values in parallel with the new cocktail. This is convenient for when on the day we switch cocktail lots, and also allows for the cocktail and assay to be set up prior to the day it goes into use. In this image, we see a snip of the BD Fact Suite library assays function where we're looking at one particular tube of an assay. Here we can go into the edit mode of the assay and assign the correct lot being used. And at this point, we create, this is in the new created cocktail for the new cocktail lot. Another route to do this would be to go inside the properties of each tube inside of an assay and to change the lot assignment there as well, and then recreate a new assay. Now we'll go over cocktail parallel QC in the work list. Once both assays for the current and new lot are in the library of fact suite, they can be run together in the same work list. This is really convenient for our lab and for the day we switch cocktails. Here you can see in the work list mode of fact suite, inside of the same work list, we're running two of the same assays, but with different cocktail lots. Both assays will be paired with the correct spillover value for the cocktail that they were stained with. This is extremely convenient also when we perform our parallel QC, that we can run in the work list mode and we don't have to go in and change the properties and select the correct lots for each individual antibody as well. For our cocktail parallel QC, we process a normal bone marrow sample, seen with both lots of cocktail, and then acquire and export the data into a layout which compares the lots. Each lot will have the correct lot-specific spillover values assigned to them, and both assays will have the same MFI targets. Once both cocktails are acquired and exported, the two runs are compared with the same gating strategy. First, we gate each leukocyte population based off CD45 and side scatter. Then we compare the two data sets against each other and our acceptance criteria. The acceptance criteria for the cocktail lot-to-lot parallel QC is less than 0.5 log decade difference in MFI between each lots of each marker in each tube. Also, less than 5% difference in population frequency between lots of each tube, and then acceptable compensation for the new lot of cocktail placed into use. All of the calculations are automated tokens which populate MFI data and pass fail criteria for each population and marker. The compensation assessment is a visual process and is not automated in the layout. Here's an example of the markers in one tube, a T-cell tube. The top 10 plots are from the current lot and the bottom 10 are from the new cocktail lot. Here we make sure scaling is the same between cocktails and visually look for comparison. The gates are linked between the top and bottom 10 plots of the different cocktails. This occurs for each tube for each assay that we cocktail in our lab. Here you can see on the histograms, the gates are adjusted to gate the positive population. On the next page, we'll see the MFI values coming from each gate, which is automatically calculated by the software, and also whether each lot passed or failed. Also documented on this is the cocktail information for the current and new lot and the specimen used for the cocktail QC. In the cocktail parallel QC layout, we have a table like this for each tube in our assay. The table looks at the antibody, in the biomarkers in each tube, and compares the MFI for each one between cocktail lots. As you can see in this example of an automatically populated table based off the previous gating in the slide before, the table has tokens which calculate the MFI difference of each marker between cocktail lots and automatically populates pass-fail. This is much faster than visually comparing the data and looking at the clicks on the plot. If all of the individual markers pass, we next check the compensation of the new lot to make sure all lot-specific spillover values were matched correctly in the software. Once the MFI of all of the populations have passed, we now look at the open gating plots where we review the new lot of cocktail only. We look at 3 pages of plots. This is just an example of one, where we assess the position of single positive and negative populations. If a comp issue is caught here, it is usually due to incorrect lot assignment in the fact suite library, or beads were used for a marker that we usually use cells for. If this occurs, a new control can be run, matched in the software, post-acquisition, and then re-exported. Once the cocktail QC passes, it is assigned off and is ready to use. The batch record is stored with the cocktail until it is placed into production and added. So our cocktail parallel QC for our screening panel is about 10 pages with 3 pages of compensation assessment. Here on these plots, we look at the single positive and the negative populations and essentially look for a straight line across. The open gating plots are really useful for this, and any compensation issue that we could possibly have in our data will be caught here before the cocktail is put into production. If any compensation issue is seen in the cocktail, it will be addressed before the cocktail is placed into use with patients on patient samples. Here we compare the same normal sample seen with 2 different cocktails. As you can see, the data is very standardized between the 2, which is the goal of the procedure we have reviewed. In this example, a normal bone marrow sample is stained with both the current and new cocktail lots. Comparable MFI between the 2 cocktails can be seen, and the comparable compensation also as well. So these cocktails are both set up with different lots of antibodies and run with different spillover values. We can see that performing this parallel and lot-specific assignment in the fact suite software allows for comparable staining patterns between different cocktail lots of assays. In order to fill the CAP compensation requirement and to assess compensation is correct, we perform the frequency of running controls every 60 days, which we have validated, and also each new lot of antibody that needs lot-specific spillover are updated as they are received. We have also validated the stability and frequency of updating compensation to 60 days and have found that it is stable even past that. We noticed that the most important updates to compensation are for new lots of tandem reagents, which we always update before use. Here in the fact suite software and the QC settings, we see that the task set for update reference settings using the universal loader and adding spillover values to the LifeSwash reference settings. You also need to choose the correct lot of CS&T beads and then have the choice to remove any antibodies that you don't want to update. We perform this process every 60 days for all of our reagents. In conclusion, reagent quality control is an integral part of high-quality data output. Lot-to-lot parallel testing and reagent qualification can help streamline the testing process and is also a regulatory requirement. Semi-automated analysis templates can streamline this QC process, and the 12-color BD-FAX lyric flow cytometer and associated BD-FAX suite application can track reagents and lot-specific spillover values, screening the use of multi-color cocktails. Thank you.
Thank you for that presentation, Robert. We have a few questions from the audience. Our first one is, what steps should we perform if an antibody parallel test doesn't pass our internal acceptance standard?
So the first thing that we do is that I look at the data and then decide whether I think that we should reset the experiment or if we need to re-titrate the antibody. Usually we find that if it doesn't pass our internal QC acceptance standard, that the new lot is brighter, and it usually results in retitration. It's not very often that we have to do it, but in those scenarios, that's what we do.
Thank you. Here's another one. How do you determine if the wash buffer solution needs to go through the 0.2 micron filter?
Well, I found in my experience that laboratories, based off of the volume that they have and the quantity of wash buffer that they make, it's kind of dependent on how long it lasts and how likely it is to have any sort of maybe bacterial contamination. I think that in my experience that running it through the 0.2 micron filter is a good quality control step to do, unless you're going through the buffer in maybe a few days, then it's probably not needed. We also store our buffer bulks in 2 to 8 degrees and only have out what we use for the day so that we can minimize the, any possible exposure. And we also keep it in like a sealed dispenser bottle as well. I think it's kind of proactive.
Okay. Are you done, or did you have more?
Yeah, I think I was just going to say it's kind of a proactive best practice to do so.
Thank you for clarifying. When titrating antibodies, why do you use stain instead of signal-to-noise ratio?
Yeah, great question. We use stain index because it takes into account the spread of the negative population instead of just the ratio of the positive and negative population. So it gives us a better indicator of the resolution that we'll have when we use that antibody in our panel. We went through the titration template and kind of that is part of initial antibody qualification before it's used in the lab. The stain index or the signal to noise ratio, I think they both work pretty well. I think the stain index is slightly better in my experience, but I think also just looking at the plots and sort of knowing the panel that you're going to use the antibody in also plays importance into the choice of the titration or the volume of antibody that you intend to use in your panel. Maybe sometimes if you double the amount of antibody, you might get a slightly better stain index, but that might not work with the volume of your reaction, or could be, excessively expensive for little return on investment. So that's all taken in consideration. Also, too, the brightness of the single signal of a particular reagent, and also what kind of maybe spillover that might lead to in your multicolor panel. So I think that although, we try to go with what gives us the best stain index, best resolution, we take all other things into consideration as well before we choose that for that final volume for our panel.
Okay, thank you. Could you review how specific antibody lot spillover values are obtained? And is this a function of the instrument software, or from FCS Express, or the fax suite?
Can I review that? Yeah, so the specific lot spillover comes from fact suite. So the spillover values are calculated in the software and like in the background. So the way that it's obtained is by running single stain controls. We discussed, we can use a few different types of controls, FC beads, which are kind of a generic control, but work for, some assays and some detectors, and then we can get more specific by using comp beads or cells staining with the actual antibody conjugate that you're using in your assay, and the exact lot, which is a best practice, especially for tandem dyes such as PE-Cy7, APCH7, some of the the BV dyes that are tandems of BV421. We do lot-specific comp for all of those. So, it's we do all of our compensation in the fact suite software. We showed, in some of the slides where the setting, the reference setting that the lot specific value is assigned to, is done in that software as well. So essentially, it's the sample is stained. You either go to the add for from function, and for most things will be added to lice wash or lice no wash settings. Choose a lot of CST beads. The software will prompt you to run your single state control. It will automatically gate that control and for beads, and then automatically calculate the spillover value, and then adjust that spillover value if it's used at different MFI settings or different tube settings for various different assays. You can also do custom reference settings, which is also done in the fact suite software, where you generate, a spillover value at a particular MFI and then save it into that assay, and it's more locked down for that particular experiment.
Thank you. To what extent do we need to quality control reagents? For instance, our lab uses LabPMM to wash samples and albumin in our final sample. Would you consider QCing the LabPMM or albumin?
So the way I don't know if in the question they're using LabPMM or albumin as part of their washing buffer. I'm just going to assume that that's one component of your wash buffer. So I would, QC the wash buffer as the final buffer that's made, not each individual component of it. That's how we do it. We create a wash buffer that uses, bovine serum, albumin, PBS, and sodium azide, so pretty standard, mixed in water at various concentrations. And we, we prepare that reagent, and then we pH it, adjust the pH as needed, and then perform a reagent QC on that. In my experience, where I've seen buffers fail is either the pH was adjusted too low, which is, I think, rare, but I've happened to see in multiple different labs that 1x PBS was used instead of 10x PBS, and it was diluted to 0.1x, and then burst all of the cells. So I think, that's one thing I don't know how that happens, if it's, the manufacturer sends the wrong reagent or it's ordered accidentally or something, but that's, every time I've seen a wash buffer fail in different labs, it's because of that. So hopefully I answered that question, but I think that, as a continuous QC program for your department, that any of the, reagents used in your panels should be, lot to lot parallel QC'd, such as antibodies and stock lysing solutions, and or QC'd, at their final reconstitution before used in patient samples, so that, any issues with your reagents can be caught before, precious patient samples could be lost, especially in, a clinical laboratory setting where maybe you have, irreplaceable bone marrow samples coming in, or, lots of samples are replaceable across many labs.
Good point, Robert. We have just a few more questions, so if you have any questions, please submit them now, and I'll ask the next one. Robert, why should we perform lot-to-lot to the cocktails if the lot-to-lot experiments in the individual antibody is acceptable?
Yeah, great question. I was thinking that I was thinking this question might come up, I mean, someone might say it's a regulatory requirement in a like CAP-CLIA clinical laboratory setting, but I won't skate out on that response. I think that you do want to perform lot-to-lot QC testing of your reagents before you cocktail them, right? Because cocktails are expensive depending upon how many tests that you're making. Even a small cocktail can be quite expensive when it's 10 or 12 colors like what we're using. And so if you verified that every vial of your like we QC every vial of antibody coming in. So even if you receive multiple vials of the same lot, I found in the past that you could have, within the same lot of an antibody reagent, you could have a bad vial. So if you don't do that, like, and you say you mix 10 vials of one reagent into a cocktail, that could be problematic, but is rare. But I think the number one reason to lot-to-lot parallel QC your cocktails is to make sure that every antibody that goes into your cocktail is actually there. So in the example we showed, in the slides, we showed a normal bone marrow sample, and for that particular experiment, or for that particular assay, a normal bone marrow sample was used for cocktail QC. That assay that we used happened to be like a screening panel. So a normal bone marrow sample provides populations for all of the markers in that panel. If a normal bone marrow sample can't be obtained, you can also, if you define in your laboratory documentation, you can also contrive a sample by spiking, say, an abnormal sample into a normal blood so that you have all of the markers inside of the parallel QC test, which is important because essentially you're looking for reactivity of all the populations that you think you put in your cocktail so that you can know that they're actually all there. Because when you go to stain, say, patient samples later on, and you have, maybe a patient who has some sort of antibody targeted therapy or B-cell depletion or something, and you don't find any, say, CD19 positive cells, you might be asking yourself, or maybe the B cells are CD20 positive or CD20 negative, CD19 positive. You might be asking yourself, was CD20 added to the cocktail, or is this biology of the sample? Same with, say, CD38 on plasma cells. So that's, for me, the number one reason. You can also, at that cocktail parallel QC, find any possible interactions between dyes that maybe a manufacturer has changed an antibody. We also look at the population percentages of the different like leukocyte populations, lymph monos, grands, blasts, hematogones, and look for any percent difference between the tubes to see if we have any reagent that maybe be killing off cells, such as maybe like CD16 on neutrophils or something, and making sure that that's at least limited and within our acceptance criteria.
Thank you. Last, how do I get more information about InVivoScribe and LabPMM's flow cytometry services?
Yeah, sure. There is information on this slide showing, so that you can definitely go to our website or email inquiry@invivoscribe.com. You could also, if any flow cytometry related questions, email me at randrews, so first initial last name, @labpmm.com. But if you don't have that written down or something, yeah, definitely our website is a good place. We have a flow cytometry section on the website that shows information on our flow cytometry panels, but also all the other gene panels and MRD panels that we do.
Thank you for joining us today, and thank you to our sponsors, BD Biosciences, for this informative presentation. Have a great rest of your day.